Welcome back to the Deep Dive. Today we are opening a file that, honestly, it completely rewrites the biography of one of the most common cells in your body. We're talking about the neutrophil.
The foot soldiers of the immune system.
Exactly. You know the drill. You get a cut, bacteria swarm in, and these neutrophils are the first ones on the scene. They rush in, they eat the bad guys,
Right.
and, well, they usually die doing it. We think of them as the suicide squad. Loyal, simple, and disposable.
Right. But the research we're digging into today shows that when it comes to cancer, that story is, well, it's dead wrong.
It's a classic betrayal story. We're looking at a huge paper by Bali and colleagues published in Cancer Cell. The title is CCL3 is produced by aged neutrophils across cancers and promotes tumor growth.
And the headline there is pretty chilling. In a tumor, your loyal defenders can become your worst enemies, active collaborators.
But before we even get to the crime, we have to talk about how the criminals were hiding in plain sight. For years, we couldn't even see these cells in the data, right?
That's the first mind-blowing part of this. To understand the betrayal, you first have to find the traitor. And our main tool for this, single-cell RNA sequencing, or sqRNA-seq, it was blind to them.
Let's break that down. sqRNA-seq is like taking a census of every cell in a tumor, right?
Yeah.
You read its diary, see what genes it's using.
Exactly. It's a census. But here's the catch. Neutrophils are genetically quiet. They have very, very low RNA content compared to, say, a big, busy cancer cell.
So they don't have a lot to say.
And because of that, the standard computer programs, the analysis pipelines, they have these quality control filters. They're designed to throw out what they think is garbage. Empty data points, bits of broken cells.
And the computer sees a quiet neutrophil and thinks, it's trash.
It thinks it's debris, precisely. It filters them out. It's like your security camera's programmed to ignore anything that isn't bright red. You're going to miss the guy in the gray coat.
You're going to miss a huge part of the population. So how did they fix this? You can't just turn the filter off and let all the actual garbage in.
No, that would just be noise. They had to be much smarter. They built something called a cell-type probability classifier. It's a really elegant piece of data science.
Okay, so instead of just measuring the volume of the signal,
It recognizes the pattern. It uses a logistic regression model. They basically train the computer to recognize the specific genetic shape of a neutrophil, even if the signal was really, really faint. And this is the crucial part. They also taught the computer what actual junk looks like. They created a mathematical debris class. So the algorithm can look at a tiny signal and ask, is this the faint signature of a quiet soldier, or is this just a piece of junk?
and the result of this new lens.
It was staggering. When they reanalyzed old data sets from head and neck cancers, they found entire armies of cells that everyone else had just deleted. In one group of esophageal cancer patients, they found 67-fold more neutrophil.
Wait, 67 times more? That's not a correction. That's a completely different reality.
It redraws the entire map of the battlefield. And of course, they had to prove this wasn't just a computer glitch. They went back to the lab bench.
Right.
They use a physical stain, CD66B, that lights up neutrophils under a microscope. And the number of cells they counted by hand matched what their new computer model found. The invisible army was real.
Okay, so step one is done. We've found the hidden soldiers. Now, step two. What are they doing? Because the paper's big claim is they aren't all the same.
This is the core biological discovery. Once they could see them all, they realized that neutrophils in a cancer patient exist on a continuum. They're not static. They change. They go on a journey.
A life cycle. From rookie to something else.
Exactly. It starts in the blood. We can call this the young recruit. Genetically, these cells are defined by a high level of a gene called SELL.
S-E-L-L.
Yep. It makes a protein, L-selectin, which is basically like Velcro. It helps the neutrophil roll along the inside of blood vessels to get where it needs to go. These young cells are naive, circulating, ready for orders.
Got it. The rookie's in the blood, Velcro on, ready to roll. Then what?
Then they get the call to enter the tumor. And that's where things start to go wrong. As they move into that toxic tumor environment, they start to change. They're pressured. They mature, or rather, they age incredibly fast.
And they lose the Velcro. The cell marker goes down.
Right. And they start expressing a whole new set of genes. This leads them to the final state, the veteran traitor.
The CCL3 high state.
That's the one. Deep inside the tumor, you find these aged cells, and they're just pumping out chemicals like CCL3, CCL4, and VEGF-A, which, by the way, helps tumors grow new blood vessels.
Okay, so why is CCL3 the one in the headline? Why is that marker so critical?
Because it's the universal flag. For years, there was so much confusion in this field. Scientists studying mice would see a bad neutrophil with a marker called Siglec-F.
Okay.
But then you'd look in humans, and that marker wasn't really there in the same way. So people would look at another one, like LOX-NOS, but that one would only show up in some cancers, not others. Everyone was using a different name tag for the bad guy.
It was inconsistent.
Completely. But Boley's team showed that this aging process, this trajectory from a young cell high cell to an old CCL3 high cell, it's the same. It's conserved across mice and humans, across lung cancer, pancreatic cancer, you name it. CCL3 is the universal fingerprint of the traitor.
A huge deal. It means we're finally all looking at the same enemy.
We have a unified target.
This feels like a good spot to just pause and take a breath. Let's do a quick recap.
Let's do it.
Recap point 1, the invisibility problem. Neutrophils have low RNA, so standard software was deleting them as debris. The fix was a new probability model that could tell the difference between a quiet cell and actual junk.
Recap point 2, the trajectory. Neutrophils aren't one thing. They age, they go from a naive circulating state marked by the gene cell into a pro-tumor age state inside the tumor marked by CCL3.
Recap point 3, universality. CCL3 is the consistent universal badge of these bad guy neutrophils, solving years of confusion between different cancer types and even between mice and humans.
But, just finding the badge isn't enough. The next question is, what is the badge for? Is CCL3 just a sign that a cell is old, or is it actually doing something?
Is it a gray hair, or is it a weapon? That's the question. And the paper's answer seems to come down to one thing. Location, location, location.
It's all about the neighborhood. The researchers use a chemical called pomonidazole. Think of it as a dye that only sticks to cells that are in really low oxygen environments.
Hypoxia.
Hypoxia. Tumors grow so fast and chaotically that they outgrow their blood supply. This creates these suffocating dead zones inside the tumor. And what they found was that the CCL3 high neutrophils were packed into these hypoxic niches.
So the environment itself is the trigger. The lack of oxygen is what corrupts them.
It forces them into a survival pact. This is maybe the most elegant mechanism in the entire paper. The hypoxia itself makes the neutrophil start producing CCL3. But here's the twist. The CCL3 chemical is a survival signal for the neutrophil itself.
Wait, hold on. The cell makes a chemical to use on itself.
Exactly. It's what we call an autocrine loop. The neutrophil pumps out CCL3. That CCL3 then binds to a receptor on its own surface called CCR1.
Like it's talking to itself.
It is. And when that key CCL3 turns that lock CCR1, it sends a powerful message inside the cell. Do not die. It blocks apoptosis, which is programmed cell death.
So it's basically making its own immortality serum.
Yeah.
I love the oxygen mask analogy here.
It's perfect. The tumor is a toxic place. There's no oxygen. A normal neutrophil would wander in, do its job, and die very quickly.
Right.
But the tumor environment teaches the neutrophil how to make its own survival kit. That kit is CCL3. The neutrophil puts on its own oxygen mask, breathes its own supply, and that keeps it alive and functional in a place where it should have died long ago.
And by staying alive, it can then work for the tumor. It can pump out other chemicals, suppress other immune cells, help the tumor grow.
That's the deal with the devil. I'll help you survive in this hellscape, but you have to work for me.
But of course, as a scientist, seeing them together isn't enough. You have to prove that one causes the other.
You have to prove causation. And this is where the paper gets technically brilliant, because neutrophils are impossible to work with. They die in a lab dish in a matter of hours. You can't just edit their genes and see what happens a week later.
So how do they do it?
Two really clever tricks. First, they did the genetic editing inside a living mouse, in vivo. They used a special mouse strain where the CRISPR gene editing machinery is only turned on in neutrophils.
Like a sniper rifle.
A genetic sniper rifle. It let them go in and delete the gene for CCL3, the mouse version, only in the neutrophils, leaving every other cell in the mouse untouched.
So they created mice whose neutrophils couldn't make the oxygen mask. What happened when they gave those mice tumors?
They implanted a really aggressive lung cancer model, and the result was stark. In the mice with the CCL3 deficient neutrophils, the tumors were tiny. They couldn't get going.
That's powerful. But they didn't stop there, right? There was another experiment.
They went one better. They needed an infinite supply of custom-built neutrophils for mixing and matching experiments. So they used something called the ER-HOX-B8 system.
Sounds complicated.
Think of it as a neutrophil factory. You can take early-stage neutrophil cells, and as long as you keep them in estrogen, they'll just divide forever. They're immortalized. This gives you time to do all the genetic editing you want.
So you can make huge batches of neutrophils without the CCL3 gene or without the CCL1 receptor.
Precisely. Then you just take away the estrogen, they turn into mature neutrophils, and you're ready for the smoking gun experiment.
Which was?
They injected tumor cells into mice, along with different types of neutrophils. Group A got the tumor plus normal neutrophils, tumor grows big. Group B gets the tumor plus neutrophils that can't make CCL3.
And the tumor growth was stunted.
Stunted. And they did it again, deleting the receptor. CCR won the ears for the signal. Same result. The neutrophils couldn't get the survival command, they died in the hypoxic zones, and the tumor failed to thrive.
That closes the loop completely. It proves it. The neutrophil's ability to make CCL3 is absolutely required for the tumor to grow properly.
It's a pillar of the whole corrupt system.
Okay, that is incredibly thorough.
Yeah.
Let's do our second recap before we bring this back to humans.
Recap point 1. The trigger. Low oxygen hypoxia in the tumor is the signal that forces neutrophils to become these aged, traitorous CCL3 high cells.
Recap point 2. The loop. The neutrophil makes CCL3, which binds to its own CCR1 receptor in a self-preservation loop, blocking cell death and letting it survive in the toxic tumor.
Recap point 3. The proof. Using amazing genetic tools, the authors showed that if you take away either the signal, CCL3, or the receiver, CCR1, from the neutrophils, you break the loop and you dramatically slow down tumor growth.
So we have solved the case in mice, but I'm not a mouse, and neither are our listeners.
Yeah.
Does this matter for a person with cancer?
This is the critical question. The so what? They took their new neutrophil spotting tool and applied it to the Cancer Genome Atlas, or TCGA. It's a giant database with genetic information from over 8,000 human patients.
So they use their classifier to hunt for this specific CCL3 high signature in thousands of real human tumors.
Exactly. And they asked a very simple question. If a patient's tumor is full of these traitor neutrophils, what does that mean for their survival?
And the prognosis.
It's grim. A high amount of this CCL3 neutrophil signature was an independent predictor of poor survival.
And independent is a very important word in statistics.
It has a lot of weight. It means this isn't just a side effect of having a big tumor or a fast-growing tumor. Even when you account for all those other factors, this signature, on its own, is a powerful indicator of a bad outcome. It stands alone as a bad omen.
Which strongly suggests these cells are driving the disease, not just along for the ride. And if they're driving, can we take away the keys?
That's the therapeutic hope. Think about it. A lot of cancer therapy is like a shotgun blast. Chemo kills everything that divides fast. That kills cancer, yes, but it also wipes out your healthy neutrophils in your bone marrow.
Leaving you vulnerable to infections.
Right. But this research points towards a precision strike. What if you could develop a drug that blocks the CCR1 receptor?
Because the healthy young recruits in your blood, the ones with the cell Velcro, they aren't using that loop, are they?
They don't need it. They're not in a hypoxic war zone. So a drug that blocks that pathway could, in theory, selectively kill the traitors inside the tumor while leaving your loyal foot soldiers untouched.
You starve the tumor of its allies without disarming your entire immune system.
It completely reframes how we think about this. We hear all the time about boosting the immune system.
Oh, all the time. Boost your immunity.
But this work shows just how naive that is. The immune system is complex. The tumor hasn't just suppressed it, it has hacked it. It has hijacked the natural aging process of a cell to build a survival suit for its own purposes.
So if you just indiscriminately boost the survival of all your immune cells,
you might just be giving the traitors a better oxygen mask. You could be helping the bad guys do their job even better.
That's a sobering thought. Precision is everything.
Context is everything. The answer was hiding in the data that everyone thought was debris.
So to bring it all together, we started with the technical problem, these invisible cells. We solved it with math. That led to a biological discovery, the CCL3 aging trajectory. We confirmed the mechanism, that hypoxia survival loop. And now we're looking at a potential new way to treat cancer.
From digital debris to a clinical driver, it's an amazing arc.
I want to leave you with a final question to think about. We always imagine our body fighting a tumor. But this research shows the tumor is using our body's own survival programs against us. So, knowing that a tumor can reprogram your own defenders this way, does it change how you view inflammation? Are we always fighting the fire, or are we sometimes just feeding the arsonist?
It's a critical question, and for anyone who wants to see that arsonist's work up close, check the show notes. We've linked to the visual abstract of that CCL3 loop. It is both elegant and terrifying.
Thanks for diving deep with us. And remember to keep questioning the data. especially the parts that people tell you are just noise.
Goodbye.